So questions on that part? Okay, so then we went on to start talking about actual new macromolecules, in particular DNA and nucleic acids, which is what we're going to continue talking about today. So the central dogma of molecular biology is that information flows one way, that it goes from the genome all the way to the proteome and out to any other macromolecules you might need to make. We know that this is not actually true anymore. There's a lot of information that argues that we actually have genetic information that proteins actually propagate, that proteins can feed back to the genome, can prevent things from getting turned on and off through post-translational modifications, et cetera, but this is a very classic way of thinking about the flow of genetic information, even though it's not completely correct anymore. And it's usually the way, when I teach, I tend to teach following the central dogma, right? Because it's an easy way to sort of follow the flow of biomolecules, go from DNA to proteins to enzymes and then on to post-translation. Okay, so past this, you should know the differences between DNA and RNA. So we spent a good long time thinking about the very, very basic structures. Right, so we're really just talking about both nucleobases and nucleosides, so ribose versus G'-deoxyribose, and how those then inform structural information about what can happen with DNA and RNA, right, and biological information. Right, so the way in which why did biology choose DNA instead of RNA? Well, the fact that DNA is far more stable than RNA probably has a lot to do with it, right? Because you don't want something that's hydrolytically labile, so something that's going to get hydrolyzed by water, to be what is your information carrier in a world where all of us are mostly water, right? That's kind of a problem. That does not bode well for long-term genetic stability. Okay, and so knowing simple things like this are really important. So these structural differences, these chemical structural differences, right? All that organic chemistry you thought you could forget actually leads into a basic understanding of the fundamental biological properties of the system. Right? And the same is true for, if you think about the bases, why we don't have uracil in DNA, because cytosine can change into uracil through immune chemistry in hydrolysis. So why does that matter? It means that you can then crop that out, right? If you know that it's not supposed to be in DNA, it's easy to see, oh, it doesn't have the methyl group, right? And although there is methyl cytosine, there is much, much, much less of that in your genome than cytosine. And so even though you can't recognize the thymine that results from methyl cytosine, right? You can recognize it before the replication fork when it's a mismatch. You can't after the replication fork when you have two single strands, right? Even though that's still a problem, and in fact you get high mutation rates there, right? It's still going to be less of a problem than if the major molecule in your DNA, if the T itself was now a U, right? So you can never tell the difference between something that can have this. Okay, so you should know what nucleobases, nucleosides, and nucleotides are. These are the bases that are attached at the glycosidic bond. These are the actual, this is the base plus the ribose, this is the base plus the ribose plus the phosphate. Okay. And again, this sort of goes back to what I said before. So you should know how receptors contribute to the biology. Okay? And so today, we are going to continue with our discussion of DNA and RNA. And we're going to go more, so we kind of covered the very, very basic molecular aspects of it, right? Just from thinking of individual bases. Now we're going to start putting these into larger scale structures and start talking about some of the structural properties of the polymers that are created, the macromolecules that are created. So before we get started, I think it's good to sort of understand just sort of what the nomenclature of DNA and RNA look like. So we've already gone through what adenine, cytosine, thymine, and guanosine are, right? So this is the A, C, T, and G that you're often going to see. Now, if you look at how they're connected, this is the 5' hydroxyl group to the 3' to the 5' to the 3' to the 5' to the 3'. When we think about how we write a DNA strand, we always write it from the 5' to the 3'. So if someone just simply says ACGT, the assumption is the adenine is on the 5' and the thymine is on the 3'. Okay, always. What does that mean? It means that this and this are two completely different sequences. Okay? This is not the same as this. You can't just reverse it. So this is the 5' thymidine. This is a 3' thymidine. Okay, and we'll see how that really affects things when we get to the duplex DNA structure. And so the genetic information is actually stored in this primary structure of DNA, right? So what this code is tells us a lot about our genes, and eventually we'll get to talk more about that. So.
Is it a typo that is facing genes or facing
It doesn't really matter. My main point was that this is not. I actually did not have this when This is a picture from the book. This I'd actually written before that for another class. So it doesn't really matter. The main point is that this does not have to match that. Although I guess if I wanted to be a little bit more precise, I could make it match. It doesn't really matter. The main point here, the main thing that you need to understand though, is that writing it this way and writing that are not the same thing. Okay. This isn't actually supposed to be like that. It just happened that they were the same. Okay. So we're going to talk a tiny bit. I don't really do huge history lessons. I think it's important to do some history of science, but I kind of try to keep it to a minimum. And we're going to talk a little bit about the history of nucleic acids. And so in 1866, this Swiss biologist, Friedrich Miescher, actually isolated DNA from salmon sperm. And so that was the first time that they actually had isolated DNA. In 1944, Avery, McBride, and McCarty showed that DNA from pathogenic bacteria could be actually transferred to non-pathogenic bacteria and make them pathogenic. So a pathogen is something that makes you sick. Non-pathogen is one that doesn't. So they showed that you could actually get DNA from one, put it in another, and actually it would now make you sick. Okay. And not only would the bacteria that they put it into make you sick, but all of its progeny, so all of its daughter cells, would also make you sick, right? So that argued that this material was something that encoded information that was then transferred to subsequent generations, right? In 1947, Erwin Chargraff reported that amounts of adenine and thymine in DNA were close to the same value. So it looked like A and T were very close to the same value. He found the same thing for cytosine and guanine, right? And this was information that then led into our understanding of what the genetic code is. So from his observation comes what we think of as Chargraff's rule. Okay, so Chargraff's rule is basically that in DNA, the total abundance of purines is equal to the total abundance of pyrimidines. So remember that purines are these bases. These have these fused heterocyclic rings, right? And pyrimidines are the ones that look a little bit like pyridine, right? They're the single, except they have two nitrogens in the ring, right? They're a single ring. Okay, and what he found was the A, so if you had a certain amount of A, you always had the same amount of T. If you had a certain amount of G, you always had the same amount of C, right? So this was a one-to-one relationship in DNA. And you can see that that pretty much holds for a wide variety of species, right? You have a one-to-one relationship between these. And you notice that if you look at a mismatch pair, if you look at A and G, that is not exactly a one-to-one relationship, except maybe in E. coli, where everything seems to be affected. But generally, it's not a one-to-one relationship, okay? So, in 1952, Alfred Hershey and Martha Chase showed that the T2 bacteriophage can inject DNA into cells, and it is sufficient to make more bacteriophage. So this is, again, this idea that DNA contains the information that you need to regenerate something. For those of you who may not know, bacteriophage is actually a bacterial virus. Okay, so there are viruses that actually grow in bacteria, and those are called bacteriophages. So viruses cannot replicate on their own. They can only replicate in a host cell. Okay, and so a bacteriophage can only replicate within a bacteria. And so they found that the DNA that a bacteriophage could inject into the bacteria was actually enough to regenerate the bacteriophage itself, which argues for genetic transfer of information. And in 1953, James Watson and Francis Crick proposed the model of the double helix of DNA. And so that was partly based on some work by Rosalind Franklin, okay, who was working in the Wilkins lab. So she had originally been promised her own laboratory, but it turned out that when she got to, I believe it was Oxford, they actually stuck her in someone else's laboratory and didn't give her an independent position. but she, with a graduate student, I believe, actually got this structure of DNA that is a crystal structure, and it shows DNA to be a helical structure. And it was shown to Watson by Wilkins, and it was actually kind of one of the things that prompted his aha moment. Okay? So, what Watson and Crick knew at the time when they were trying to solve the structure of DNA, they knew Chargaff's rule, so this idea that you have this one-to-one pyrimidine to purine, right, and that A's and T's seem to be similar and G's and C's seem to be similar, that argued for some sort of pairing, right? And they knew that DNA had to be a helix from Rosalind Franklin's work. And we're going to go over the structure in excruciating detail. But before we get to that, I actually have a little bit of a side note, which is, this is kind of like a word on scientific hero. And it's really a word on James Watson. And so, you know, we talk a lot about Watson and Crick, and Watson had, you know, and he was a brilliant person, and he and Crick did solve the structure of DNA. But he sold his Nobel Prize in 2014, and he was widely criticized for his sexist and racist remarks. And before you say, oh, but this is just someone else's say-so, I actually went, when I was an assistant professor, Watson came and gave a talk at UT Austin, and it was this big symposium. And I walked out of that going, oh, goodness. It was, you know, it was a sight. That's all I have to say. You know, it was not the kind of thing that you walked away from thinking, wow, that's somebody who I want to be my scientific hero. So why am I pointing this out? The reason I'm pointing this out is because I just want to point out that smart, famous people are not always right. Okay? Just because they're smart and famous does not mean that they are always correct, you know, or good role models for how you should be. Okay? So I just want to point out that just because someone wins a Nobel Prize does not make them a good human being. And however, you know, it's a point, right? Because we have this tendency, right, to put people on pedestals, and we think, oh, wow, they're so smart, they're so wonderful. But smart and being like what I consider to be, you know, somebody who I would prefer as a scientific role model, they don't necessarily go hand in hand. And I've met many Nobel laureates. In fact, I worked for a Nobel laureate. I've had a lot of really wonderful people who are Nobel laureates, but not all of them are, you know? And a lot of them, not a lot of them, but a significant fraction of them, you know, can sometimes do things that you really wish that they didn't. So, but on a happier note, to talk about Rosalind Franklin, she was actually a female chemist who was responsible for a lot of the research leading to DNA structure. Really, that was such a critical, that alpha helical formation, the helix, the dual helix formation that she got, so that crystal was so incredibly important in terms of solving DNA structure. And in fact, Wilkins, whose lab she was in, actually won the Nobel Prize with Watson and Crick, based on her work. And let me say that they actually, it was her article on the cryptographic data that was in the same journal as Watson and Crick. And in fact, she was pretty close to solving it when their paper came out. Now, to be fair, she presented her work at an earlier place, so to some extent, you know, she'd already put her work out there, and she was never very resentful about the fact that this had happened. She died before the Nobel Prizes were awarded, and they do not give them posthumously, so that's part of why she didn't get one. So it's not exactly a case of scientific misconduct. This isn't a case of scientific misconduct. The reason that people are usually unhappy with, especially, because apparently Crick and Rosalind Franklin were actually friends, but part of the reason people are unhappy, especially with James Watson's treatment, is because he wrote a book in which he talked all about the glorious ability they had to solve the structure of DNA and completely minimized their contributions, except in an epilogue. So it was kind of one of those things of, you know, if he had just been a more magnanimous person, people probably wouldn't have backlash against some of them. But that kind of goes with why he sold his Nobel Prize. So, okay. That said, let's talk about actual biochemistry. So this is, you know, what we think of as the B form of DNA. This is the classical canonical double helix. We're going to talk about it a lot. So what are the key features of this Watson-Crick model? Okay, first, it's a right-handed helix. So if you look at the way in which it actually, if you follow the helical curve, the axis goes through the right hand, so it's a right-handed helix. It is anti-parallel, so we'll talk about this more in a minute, but what that means is that it goes 5' to 3' on one strand with 5' to 3' on the other strand, right? So the strands are running in opposite directions. The bases are on the inside, right? So all of the bases are stacked on the inside of the helix. Okay. The original model before they saw Franklin's structure was actually a weird 3-strand helix. Okay, that did not make as much sense. And then after they saw the new structure, it made a lot more sense to do this dual-strand Chargaff rules. The phosphates are all on the outside, which actually makes sense in terms of minimizing charge repulsion, right? Lots of negative charges don't like to be in the same space. And there's a complementarity of bases, right? Okay, we're going to go over these key features. So, getting to this idea of the anti-parallel right-handed helix, to really bring that home, if this is the 5' strand, if this is the coding strand, right here, here's the 5' strand here, which I have a sample bit in orange here, and it is complex with an anti-parallel strand that's running 5' to 3' in the opposite direction. Okay, so these are this way, right? They're anti-parallel to one another. If both of my hands are a strand, and this is the 5' of one, and this is the 3' of the other, and 5' of one, and 3' of the other, they're doing this. Okay? Okay. And you can see from this, and this is just another example, so here's, 5' to 3', 5' to 3', and you can see that the 3' here, so what's coming off here is actually complexed with the base on the other side of the strand, right? This is reverse complementation. So this is the reverse complement. We'll talk about that more in a minute. Okay, so another key finding by Watson-Crick was figuring out what we now call Watson-Crick base pairing. So this idea of what goes with what and how, okay, which is really, really critical. So in those bases that are running in the center of that DNA molecule, right, that are where you have the reverse complementarity, okay, you have an adenine with a thymine. So you can see you have 2 hydrogen bonds here. And you have a guanosine with a cytosine. So this is the famous ATGC base pair. And here you have 3 hydrogen bonds, right? So this one, the adenine is a hydrogen bond donor, hydrogen bond acceptor. Here it's a hydrogen bond acceptor, hydrogen bond donor. Okay? If you look the same here, hydrogen bond donor, acceptor, donor, acceptor, donor, acceptor, right? So 3 here and 2 here. So here's a question. Well, actually, I already answered the question. What holds together the Watson-Crick base pair? So what do you think the role of this is in DNA? What's the role of Watson-Crick base pairing in DNA? There are a couple of different roles it could have, right? So one thing that I should point out is obviously these complementarity is very important, right? So you have a very, very clear coupling from your 5' to 3' on one to the 5' to the 3' on the other. You're having complementation. Why would that matter? What's important about that?
It kind of gives you some foundation for DNA replication.
It does. It gives you a foundation for replication, but why? It's specificity, right? These hydrogen bonds are very, very specific. Okay? If you try to base pair A with C, you only get 2 hydrogen bonds, even though this can accommodate 3, right? If you try to base pair T with G, the same thing happens, right? You can't get a perfect hydrogen bonding match. And that leads to specificity in the code, okay? And it's a recognition event in the code. And that actually very much goes with the idea of replication, right? So that when you have a choice, if you have an A, when you have a choice between a T or a C or a G, right, only the one that has the proper orientation of the hydrogen bonds will fit in opposite to it, allowing you to replicate again and again and get always the same thing on the opposite side. Okay, so that's what this is really towards, is the specificity of the genetic code. So, and we're going to talk about this again, when we just talked generally about the importance of non-covalent interactions in nucleic acid structures. So this applies to the helical DNA that we're going to talk about, but it also applies to the RNA DNA and RNA molecules as well. Okay, so these non-covalent interactions are at play in all of them. So there are really kind of, and we've sort of discussed this at the beginning when we first talked about non-covalent interactions. I introduced DNA as one of the examples that I've used, right? So there are a couple of different non-covalent interactions that one can think about in DNA, but really there are two main ones. I mean, the third one is just the metal ions shielding the backbone from repulsion. Why is that important? So you have magnesium that usually binds all along the phosphate backbone. It helps to prevent charge-charge repulsion. And it's important because magnesium is actually really, really critical in our biology as a result. This is part of why we need so much magnesium, because because it's what we use biologically to help shield our DNA. Okay, it actually complexes very nicely between the phosphates. And so you often see magnesium bound in DNA structures, right? Okay, but besides that, which we're not really going to talk about because it's not a very specific interaction, unless you're really interested in bioinorganic science, which actually, let me just say, my colleague Daniela Bucella in the chemistry department is actually looking at pools of magnesium in the cell and trying to understand how it might influence cell signaling, right? And thinking about bound and unbound pools. So there's a lot of biology even here that is still to be discovered. But for our discussions of DNA, we don't really need to think about this that much other than just a general charge repulsion prevention agent. But the two things that we do need to think about are hydrogen bonding between the bases, which, as I talked about before, gives you specificity, and aromatic interactions, which is really one of the main thermodynamic driving forces, okay, of complementation, of the helical formation. So hydrogen bonds, again, it's not really the thermodynamic driving force. I'm not going to say they don't matter at all. There is a little bit of thermodynamic stability given by hydrogen bonds. Not very much, about 2 to 6 kilojoules per mole, right? So very, very low. And that's the real number when compared with hydrogen bonding to water. Okay, so the 2 to 6 is the real number, including the competition. So it's really not the main thermodynamic driving force of helical formation. However, they are very, very critical for specificity, right? It's really important that A couples with T and G couples with C, okay? It is important for specificity, and it's also cooperative. So if you think about the way that nucleic acids actually fold, they fold cooperatively. What does that mean? They start at the bottom and they kind of zip up, or wherever they meet, they zip up and down. Okay, and that's really a probability if you think about it, because remember these hydrogen bonds are directional. They are an orbital overlap, right? So they have to get in just the right position to have the strongest hydrogen bonds. So once you have the first one there, it now holds the other two closer together and increases the probability that they'll snap together, increases the probability that the next one will snap together, increases the probability that the next one snaps together, etc. And so therefore it's a cooperative interaction, right? So it enhances, so having the second one enhances the third one, enhances the fourth one, and on and on and on until you zip it all up. Okay. Actually, this has an impact on how we think about the thermodynamics of going from a helical structure to a single-stranded structure, right, as we talk about melt temperatures, which we'll get to a little bit later. Okay. So, again, I know I'm being very, very repetitive, but I figure this is an important point to really get home. So again, A gives the T or U. So the hydrogen bonding patterns of uracil in RNA is the same as the hydrogen bonding patterns of thymine in DNA. Remember that the only difference between those two is this methyl group here, right, which is outside of the base-base interaction, right? So this does not impact the Watson-Crick base pair. And GC. So if you look at AT and GC, one of the things that we find is that there are 3 hydrogen bonds for GC and 2 for AT, and that does add a little bit to the stability and to making GC base pairs more thermodynamically stable, so they hold the DNA together a little bit better than the AT, okay? It is one of the factors, and we'll talk about the other in a minute. It is not the only factor that leads to GC being a stronger interaction than AT. In terms of thermodynamic stability. So again, it's really critical for specificity. It does contribute to the stability, but not very much, right? So it's about 2 to 6 kilojoules per mole per hydrogen bond when accounting for water, but it's not as important as base bond. So, getting back to this whole issue of the genetic code and the fact that you have this pairing, right? I just want to point out something that I sort of said, but I didn't really have a slide on, that this right here, if we think about when we write a DNA sequence, right, we write it from 5' to 3'. We often call the 5' to 3' the coding strand, right? And this would be the non-coding strand. So if we're thinking, so what I'm saying is we would say that this direction, if we had a piece of DNA that was coding for the genetic code, right? Whatever we typically write from 5' to 3' from our sequencing data, where we sequence the genes in something, is the coding strand. And the opposite is the non-coding strand. So this is not just the opposite, it is a reverse complement. So the 5' is over here. If we were to write this in the same direction as that, we'd start with a C. Okay? So oftentimes we shorthand a DNA duplex by simply writing down the coding strand. And the assumption is there is a reverse complement, reverse complement DNA strand going the opposite direction that is hydrogen bonded to it, okay, to make the DNA duplex. So if I say this is the coding strand of a piece of DNA, you would assume it's duplex and there's another strand going the opposite direction. And if I were to say write reverse complement, right, or tell me if the duplex that this would encode is higher or lower in thermodynamic stability, right? What you would know is that there's a reverse complement strand going the other direction. Okay? Okay, so why am I talking about coding and non-coding strands, and what is it code for? Questions? So when we talk about, this is a good question, when we talk about the thermodynamic stability of DNA, what we're usually talking about is the melting temperature between duplex DNA, where it is in a helix, where both the coding and non-coding are present, and the two independent single strands. So we are not normally talking about the thermodynamic stability of the individual strands, right? So because we're really talking about like what the non-covalent interactions are bringing to the thermodynamic stability. So we're usually talking about the stability of the structure. If there were other secondary structure elements that were single-stranded structures, you could talk about differences in them, right? But generally, the base stacking is not, well, generally I think the base stacking is still not exactly the same. So if you were talking about them as independent single strands, they wouldn't necessarily be the same. But that's not typically what we mean when we're talking about the stability. We're usually talking about the stability of the Okay, so part of why I'm getting into this coding and non-coding nomenclature has to do with this genetic code, right? So I wanted to just point out that DNA is a carrier of genetic information, okay? And in your parent molecule, so if you have your parent DNA molecule, right, this actually unwinds into two independent strands, each of which encodes fully a new copy of duplex DNA, right? Because, so we have sort of conventions that guide what we consider the coding strands. And it's not always, because of the way that DNA encodes genetic information, especially encoding RNA and proteins, it's not always obvious. Sometimes the way we think about it, it's actually the reverse complement strand. So there are genes that you can have genes that are sort of overlapping and on different strands. And that gets much more complicated than we're going to talk about now. Hopefully we'll have time at the end of the semester to start talking about some of that in the context of transcription and translation as we start to think about how these molecules are replicated and what the biochemistry of that is. But my main point here is that your parent DNA molecule, because of this Watson-Crick base pairing specificity, right, actually has all of the information to create a new strand that's exactly like the old strand. So you notice this strand here, which is brown, is from the parent. This blue strand directly replicates that brown strand, right? So it's a direct replication. So each strand is acting as a template for a new complementary strand of DNA, right? To replicate the DNA. And this is actually what we call a replication fork. So when I was talking about the replication fork, if a mismatch pair is before this point, right? And you can tell that they're different. You can actually catch it for that thymidine. But if it's past this point, how could it possibly tell that it's not the original, right? So that's why you get those mutations.
I think like thermal dynamicity, if you have a detached like strand DNA that has
We
more
are going to go into thermodynamic stability in like exquisite detail. So I mainly stopped here for a minute just because we're talking about hydrogen bonding, and this is a good place to talk about this. Since I'm not going to go into detail about this, and it's kind of an aside. I just wanted to point out that this is the specificity of the genetic code. But we're going to have a very long conversation about exactly that. So, okay. Okay. So getting back to non-covalent interactions and how they influence things like thermodynamic stability, the dominant driving force in thermodynamics is actually base stacking. Okay. So remember that when you think about nucleobases stacking, this distance is very short, right? And they stack really right on top of one another. These are two aromatic surfaces that actually have permanent dipoles as well. So it's not just the aromaticity, right? It's not just the transient van der Waals. There's actually dipole-dipole interactions involved that are one on top of the other. And so you see this even in non-base pair regions. So this is an example of an RNA, right? This isn't actually a DNA structure, it's an RNA structure. And you can see that this RNA structure has a helical section where you can see that they're stacked one on top of another within the helix. But it also has a non-helical section where these aren't base paired to one another. And yet you still have stacking, right? So even in parts that are not base paired, you still have that aromatic interaction. Okay. And if you look at the B form of DNA, and we're going to go through, so this is the classic Watson-Crick helix. It has a very specific name. It's called the B form of DNA, and we're going to go over it exactly what its properties are in a minute. If you look at the B form of DNA, and you look directly down the axis of a Watson-Crick helix, you'll see that you have your bases slightly canted to one another. They're all stacked, and they turn just a little bit, and you can see them being stacked all the way down. Okay? Okay, so as I said, these aren't just a matter of aromatic interactions. These aren't just aromatic molecules. They actually have dipoles. So if you remember from the last lecture, this is the electronegativity map of those, right? Where these are, where the blue is less electronegative or electron deficient, and the red is electron rich, kind of a classic organic chemistry rendering of this. And what you see is that these all have different surfaces, right? They have different dipolar surfaces. Why am I pointing this out? I'm pointing this out because the stacking of an A over a G versus an A over a T, right, or an A over an A is different, right? Those interactions are going to be different. And you do not need to memorize these numbers. So let me point out, I'm putting these here just for your reference. Okay, these are not numbers you need to memorize. But my point here, the reason I put this here is that this is actually based on multiple different data sets, changes in the enthalpy, entropy, and free energy of independent sort of structures that have either a G over a C, right, or a G over an A, or an A over a G, et cetera, et cetera. And what you can see is, if you have a GC versus a CG, there are small differences in the delta G of how favorable it is, right? If you have a GG versus a CG, there are small differences, right? This is actually less favorable than the mix. Okay, so this stacked over that is more favorable than this, this stacked over that is more favorable than this stacked over itself. Okay, and you notice that if you have an AT, it's a lot less favorable. So an AA, so A stacked over A, or an A stacked over a T, it's a lot less favorable than a GC. So when we talk about the fact that GC interactions are more favorable and actually contribute more heavily to lead to more thermodynamically stable helices, it's not just because they have 3 hydrogen bonds. It's actually because the dipole-dipole interactions of their aromatic surfaces are actually stronger as well. Okay, so it's 2 things. It's that thermodynamic stability is led not just by the hydrogen bonding, but also by this stacking interaction.
Does that make certain DNA mutations more common than others?
It's not so much a matter of, so it affects a lot of different things. I don't think it's a matter of mutation. It's more a matter of the stability actually changes your ability to unwind certain pieces of DNA. So how they then, how accessible they are, how much energy has to go into it, how stable they are, definitely is different. I think the mutational aspects of it tend to be more things like when you have hydrolysis or DNA damage or other things of that nature. So those are usually editing mechanism problems, which again, if we have a little time at the end of the semester, I'll go into a little more. Okay, but my main point here that I really wanted to drive home is that even within a sequence, you could have the same numbers of A's, C's, G's, and T's, and have two sequences that have different thermodynamic stabilities, right? So it's not just the percentage of G and C that leads to thermodynamic stability, right? Because the sequence of what stacks on what is actually important in that stability. And again, you don't need to memorize it. This is the take-home. Okay, I just wanted to show you that because I feel like this is a nice way of actually putting that. Okay. So in thinking about, so what I've been talking about were sort of the general non-covalent interactions and how they sort of lead to structural information, both in terms of thermodynamic stability of both DNA and also RNA structures, okay, because except for the T, I mean, really is about both of them. But also, but how do they come together in what's sort of the classical double helix, right? So the classic double helix is a very specific structure. It's called the B form of DNA, okay? The B form, why it's called the B form, I don't know. It was named the B form. And if you're asking, yes, there is an A form, and we're going to talk about that too. Okay? But the B form of DNA is actually of DNA. RNA cannot attain the B form. And we'll talk about why that is in a minute. The B form of DNA has a couple of very specific things about it. It has, obviously, the anti-parallel helices. One turn of the helix is 10 bases. Okay, so that's there are 10 bases in a turn of the helix, and there's a 34 angstroms from turn to turn. Okay, in a full turn of the helix. There is a groove. There are two grooves of DNA. There is a Y groove that's called the major groove, and if you look, there's a more narrow groove that's called the minor groove, and that's running along here. Okay, the major groove is about twice as wide as the minor groove in DNA. Okay, the major and the minor grooves have, if you go back to the Watson-Crick base pair initiative, you'll notice that there's a thing on the top that says major groove and a thing on the bottom that says minor groove, and you'll notice there are different hydrogen bonding patterns for the different base pairs that stick out into the major and the minor grooves. Okay, and that is actually important in recognition. I'm not really going to go over that right now because, again, if we manage to get some time for talking about some of the transcription factors and how they act and all of the rest, we'll talk about that more. But I will say that proteins do tend to recognize DNA more in the major groove than in the minor groove, perhaps for obvious reasons because it's wider, there's more room for binding in that particular group. And there was a question. So if you follow the curve of the helix, if this thumb's up, it's right-handed. If it's curving this way, it's left-handed. So this is actually curving that way. Like if you look, the curve is going this way. It's curving like. It's really hard. But if you look at the left-handed, it sometimes becomes more obvious. I have to admit, I too have a problem. Like you have to kind of look at the curve of the helix, and if you follow the curve. This one's a right-handed, and the other is a. We'll talk about the left-handed. Yeah, it's a counterclockwise. Yeah. So I have to admit, it's kind of one of the harder things to see. Yeah, like something that's curving this way is right-handed. Something that's curving this way. So these are opposite. Right is left-handed. Okay. Okay. Well, we'll work on that. But in the meantime. One of the things I also want to point out while we're on the subject of just numbers, right, sort of trying to understand what some of the numbers are for this, is that the distance between these base pairs is about 34.34 nanometers, right? Okay, so that's the distance, again, between these two base pairs, which is, I think, yeah. Between the two base pairs that are stacked. So there's a very, very small gap between the two base pairs, right? So two base pairs. The main thing you need to know is not that number, but rather that they are very close together. Okay, that's the critical aspect of it. And that's important because this is this is a dipolar and sort of van der Waals-like interaction. The pi-pi stacking is kind of a combination of permanent dipole and van der Waals interaction, right? And that's distance dependent. They have to be really close for that to have any energetic, to have any real energy to it, right? For it to have any real meaning in terms of being a thermodynamic driving force. I think it's actually pretty uniform. I'm sure that everything breathes, right? So I'm sure that as you get, that for some of them there may be slight differences. And let me say that nothing is perfect, right? So if you actually look at crystal structures of real DNA, they're slightly off. They have slight kinks in different places, and they're slightly canted, right? So this is the idealized model of DNA. And it roughly follows what we actually see, but it's not perfect, right? DNA structures aren't perfect in reality. Okay, so these are kind of the numbers that we give for sort of like idealized perfect conditions. But I do think you're right. So there are probably slightly different van der Waals radii for the different bases. It's not enough to make it significantly different for us to worry about. Okay, so going back to something that you may not remember that we talked about in the previous time. So we talked about how 2' deoxyribose can adopt this C2' endo conformation. And just to remind you, this is the envelope form of basic, remember like cyclopentane, the envelope form? So this is like ribose, it's like a 5-membered ring like cyclopentane, so it's the envelope form. So this is coming up. This is the C2, right? This is where that hydroxyl group would be in RNA. You notice it's really, really, really close to the phosphate, so that hydroxyl can't fit, okay? That means that this conformation cannot be adopted by RNA. The reason that's important is because this is the conformation that is required for getting the B form of the helix, okay, for getting the B helix. So the B helix requires the C2' endo form of ribose. So in order to get this exact helical conformation. So only DNA, not RNA, will form B-form helices, okay? So B-form nucleic acid helices are reserved for DNA. And in fact, the majority of DNA is found in this type of helices, okay? Okay, and just getting back again to, if you look at the B-form helix, again, this is the view down the helix. So we saw this before. You can see that the bases are slightly canted. These gray rectangles this is actually meant to be a base. And they're centered, in the B form, they're centered down the axis, right? It's like a straight shot down the axis. If you put a pin through the middle of where the hydrogen bonding of the two bases are, right? It's completely centered evenly through the axis of the molecule. On average, obviously, slight variations in reality, right? So, and the planes are perpendicular to the axis, so they're all stacked like this. Right? They're stacked one on top of the other. And they're slightly true. Okay. Okay. Now, that's the basics of the B form helices. What about the A form, right? Like, so we got to B before we got to A, because B is the more common for DNA. The A form helix is actually commonly found in RNA DNA and RNA-RNA. You very rarely find it in DNA. The only time it's actually found is if you dehydrate DNA, and so it's usually considered an artifact of DNA preparation. Okay, so DNA helices in general in physiological solution don't form the A form of DNA. What's different about the A form? So like, what is the A form of DNA? The A form of DNA is a lot like the B form, but it has some slight differences. It's more compact, and you can see that here. This is one turn of the helix. You notice that this is kind of squatter, right? Like it's a little bit squatter, it's got a few more, it's actually like, it looks kind of fatter, right? It's kind of shorter and fatter, basically. And it has 11 bases per turn instead of okay? The bases are tilted. So if you look, here these are completely sort of, evenly distributed straight on the axis. These bases, they're a little off, right? They're a tiny bit tilted, okay? They're not, and they're also not centered down the axis. Like if you were to look down this helix, the bases are not centered down the axis the way that they are in the A form, and you can kind of see that here. Right? If you go straight down, you notice that I'm not going through the middle of everything the way I am here. Right? So one of the reasons that you form an A form and not a B form is now and you don't really need to know exactly what this looks like, but it's a different envelope conformation for ribose, right? So now the C3' is endo, which means coming up, not the C2'. So it's a C3' endo for ribose for the A form of DNA. Okay, and so that is a conformation that RNA can adopt within the structure, within the nucleic structure. So that is why you get the A form. So again, this is what you see in RNA DNA helices and RNA DNA and RNA helices.
So you are making that information how we do that.
DNA can only form, so this is not, in physiological solution, DNA will naturally go to a B form helix. The A form is only seen for DNA under conditions of dehydration, which argues that thermodynamically, which it kind of makes sense actually from just the stacking interactions by itself, right? Thermodynamically, the B form helix is more stable than the A form helix. It's only when it's sort of forced to that you'll end up in a B form. That you will end up in an A form for DNA. So, in terms of, naturally, I think DNA, as part of the replication process, the structure will change in the immediate vicinity. And so, I suspect that at some point, some natural DNA may adopt a B form as part of the replication. Actually, probably not because you're unwinding, right? You'd really have to wind more. But there are probably cases where B form is natural. Generally, though, when people look at DNA structure, they do not see a lot of examples of physiologically relevant A form DNA. They almost always see the A form structure when they look at helices that have RNA in them. So DNA RNA, because of the fact that it can't form that C2' endo, or RNA RNA. So, I mean, I suspect it's because thermodynamically it's not the most stable, and so it tends not to adopt that as its preferred helices. But, in nature, you see a little bit of everything, is the reality, including things that we don't really understand yet. No, it can. In fact, it does. So that's why it can do RNA DNA. But I think the real reason that it doesn't do it is because if you think about the stability of an A form versus the stability of a B form, here you have all of your bases, right? Like perfectly stacked upon one another going down that axis. Here, they're canted, right? Like everything, if you look, they're canted towards one another. So they're not you don't have as much of a line stacking interaction. And I think that that's part of it. So I think that you don't get as good of an aromatic interaction in the B form. In the A form as you do in B. Sorry, it's confusing. In the A form of the helices. I won't call it DNA because you don't see the A form of DNA. The A form of DNA is known to be an artifact of isolation, like when people add alcohol. So. Yeah, so actually DNA can indeed adopt this, which is why you can see it in RNA DNA helices, and that's exactly what you do see, actually. Okay, actually direct comparison of the two. You can kind of see it more obviously here. You can see that they're not perfectly, so here they're all perfectly perpendicularly stacked. Here, they're not, right? And they're both right helices. There are 11 residues per turn in the A form, there are 10 in the B form, so this is a more compact form of DNA, right, from dehydration. For DNA, they're actually showing a DNA, so they're showing the dehydration impact. And this is a little bit more squat, right, so if you look at it's wound a little tighter, it's a little shorter, right, it's a little bit more squat, and all of the things are slightly up. Okay. So I just want to take a minute to talk about syn versus anti-conformation since we hadn't covered that before. So if we think about the nucleobases, if we look at deoxyadenosine or any of the purines, This purine can be in two conformations. It can be so that the aromatic ring is over the ribose, right, in which case it's called the syn form, or where the aromatic ring is away from the ribose, in which case it's called the antiform. Which do you think is more stable? I want all of you to take out your little extra credits if you have them, and tell me which of these two is more stable. For the purine. And then tell me if you think the same is true for the purine. And I will warn you that these aren't drawn to scale, right? Like everything's not exactly the right size of tissue. Yes. At the attempt of why. Which do you think is stable, and like one reason why you think it might be more stable? You know? You don't have to be right. I just kind of want to see what you guys are thinking, right? These are thought exercises. They're good for you. You know? Okay. So the anti-conformation in both cases is the more stable conformation. Does anybody know why?
There's steric hindrance in the same conformation because everything's on the same side.
Yep. So that's definitely true and obvious for the purines where you have steric hindrance. Here, why is it true for the pyrimidines? Yeah, so it's actually also a matter of sterics, but it's also electrostatics, right? You have to remember this oxygen has lone pairs that are now clashing with the lone pairs on the oxygen of the ring, right? And so there's repulsion between the two lone pairs, which doesn't happen here. Okay? So it is absolutely a matter of sterics. So, and electrostatic repulsion in the case of the oxygen-oxygen. Right? So you do have this hindrance, and so it is true that the anti is the more stable form. And in fact, in both the A and B form conformations, so B form DNA, which is our standard, and the A form, which you see again, a dehydrated DNA or DNA, RNA, helices, all of the bases are in the anti-conformation. So part of why I'm introducing this to you is because there is a weird form of DNA where this is not true. Okay, and it's called the Z form of DNA. I know, like A, B, and we went all the way to Z. If you ask me if there's anything in the middle, no, not that I know of. I think it's called Z because people thought it looked like a zipper or a zigzag. Okay, it's actually a left-handed helix instead of a right-handed helix. So now if you curve around, this is actually curving this way. Okay, this is an example of a left-handed helix. I will say that although there are now definitely confirmed examples of zDNA in biological systems, nobody knows what it does. So talking about mysteries, right? It's restricted to DNA, so this form is only seen in DNA. We have absolutely no idea what it actually does in DNA, like if it has some specific genetic structural event, right? Like if it controls something, if it's made for a purpose, we don't know. It's a mystery. We know that in the Z form, the purines, so those big molecules that, the double ring, right? They're actually syn. They're in towards the ribose. Whereas the pyrimidines, which I think because they're smaller and the Z form can still accommodate them, are anti. Okay? And it is a C2' endo sugar, which explains why you only see it for DNA and not for RNA.
Is there any reason to see
Not that I know of in DNA. It's possible because RNA, as we'll get to, adopts a really wide variety of structures that you might see it in RNA. But in DNA, not, in double-stranded DNA, not that I know. Okay. So I want to point out that DNA can form higher order, or what we now consider. So all of the helices we consider those secondary structures. DNA can form higher order or tertiary structures. So holiday junctions are one example of that. Another example of that are supercoiled DNA, where you now have coils of coil, right? DNA can differ in size and shape. They can be, in this case, this is an example of circularized DNA. Like plasmids, which we use in molecular biology, are another example of circularized DNA. It can also be linear. So we can have a linear circularized DNA. It can be relaxed. It can be supercoiled. I'm not going to go into all of the dynamics of supercoiling. I know the book does. You notice I didn't assign you those chapters. Okay. But there are many different forms that DNA can take. However, the vast majority of DNA, if it's in a duplex, is in the B form. Okay, even here it's in the B form, but now you're getting higher order architectures. This is important because, as we'll get to hopefully later in the semester, the supercoiling of DNA is part of what leads to its packaging in things like chromatin, right? Where you have to have coils upon coils. If you actually think about all the DNA in our chromosomes, we have an insane amount of DNA in a very, 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 very tiny space, and that's because it's packaged incredibly well. Okay, but that involves a whole regulatory machinery that we are not at right now. Right? We're still learning about basic molecules, so we're gonna not go into that yet. Okay, so that was really the helices that we were talking about. I mean, as I said, the A form we see for RNA, but let's talk a little bit about the kind of tertiary structures that RNA can involve. Right? So DNA is all helices and then twisting of helices and helices and helices, right? But RNA can do some pretty amazing things. This is just a sampling of different forms of RNA structures. Okay? This is what's called the hammerhead ribozyme. It was the first RNA enzyme that people really had identified. This is an example of a hairpin. This is the hepatitis delta virus RNA. And this is a group 1 intron, which actually is something that happens to do with splicing, which we'll get into much, much later if we get over there. Okay. And this, just in case you didn't know it, is the ribosome. And we often think of the ribosome, which makes all of our proteins, as being mostly protein, right? Yeah, not so much. It's mostly RNA. Okay? The ribosome actually, this is work that Tom Seitz and others at Adionic won the Nobel Prize for, right? The ribosome is actually a ribozyme. It's a catalytic RNA. Okay, it's an RNA that can act as a catalyst. It's a pretty amazing molecular machine, has incredibly complex folding. And the vast majority of what we consider the ribosome is RNA and not protein. Like this is the protein, this purple part's the protein. All of that gray stuff is the RNA. Okay, so RNA can do some crazily amazing things. It isn't just limited to that helix, right? And so just to sort of bring that home, so this is actually a transfer RNA, right, which we'll get into if we get to translation. So this is, if you look at transfer RNA, this is what we would consider the primary structure when you just write it all out, right? Looks kind of simple. This is actually the secondary structure. This is what it folds into. And so you can really see the secondary, we look at this as a secondary, but then if you really look at it, it forms this tertiary structure, right? It's a pretty amazing, complex fold. So I just want to point out that proteins often recognize DNA sequence. So when a protein is recognizing DNA, it's usually just by sequence. With RNA, the structure is so important that proteins often recognize not only the sequence of the RNA, but the structure of the RNA, right? That's because DNA, normally B-form helix, right? RNA, amazingly diverse levels of structure, okay? And this is really, really important in biology. We still don't understand all the ways in which these RNA folds can influence everything from regulation of translation through the ribosome to regulation of the stability of transcripts of mRNA, to the use of small molecule, or not really small molecule, but small RNAs called microRNAs that can then actually cause degradation of RNAs through other machineries, right? There's a huge, huge biology now that's really developing along the fact that these molecules are incredibly diverse in what they can do, okay? Which is why, even though this is not in your book, I really thought it was very important to go over it, okay? So I just want to point out that, we talked about Watson-Crick base pairs, And those are the canonical base pairs we see in DNA, and you see those in RNA as well. Okay? But RNA oftentimes also make use of what are called Hookestein base pairs, which are these base pairs here. Okay? So in a Hookestein base pair, it is a protonated cytosine that can actually base pair with a guanosine, which is already base paired in a Watson-Crick with another cytosine, right? So you can actually get these bridge structures. And here, in a Hookestein base pair, a uracil can actually base pair with an adenine. Okay, again, making use of the other face of hydrogen bonding, okay, of these nucleobases. Okay, so these are some of the common secondary structural elements of RNA. You don't really need to know what all of them are, but you notice that there are a wide variety. We're not just in helix land, right? And so whenever you have the lines going across, this means that these have Watson-Crick base pairs, or they have hydrogen bonding, okay? And you can see you can get single nucleotide mismatches where now you have what's called a bulge. You can have something very common where you have mismatches where you have a stem where all of these are hydrogen bonded, and then you have a region that has no hydrogen bonding, right? And that's called a hairpin loop. It's a pretty common structure. You can get to more complicated things like this asymmetric internal loop, these different junctions. You can make all sorts of different structures with this. Again, this is probably one of the more common structures, and you don't need to have any of this memorized. I just wanted you to get a sense of the variety of structures that RNA can adopt. And you can get tertiary structures. This is an example of a pseudoknot, where you have base pairing here, no base pairing here, base pairing here, no base pairing here, right? So it looks like a knot. I like this one, it's called a kissing hairpin, right? It's like those two hairpins, but now they're connected to one another, right? So it's a kissing hairpin. I think it's a cute name. And this is a hairpin loop bulge contact. Again, you don't need to know the specifics. My main point is that these things can actually form pretty complex architectures through base pairing, right, through appropriate hydrogen bonding interactions. Okay, and the last point I'm going to make about RNA is that one of the reasons that people are really interested in RNA, besides the fact that it turns out that they're really critical to genetic regulation, right, is that there is an old hypothesis that RNA actually probably came before DNA, which makes sense because you would probably have ribose before deoxyribose, right? And that actually the fact that RNA can be catalytic and the fact that it can encode information as it does in translation may mean that maybe RNA is actually the precursor to the self-replicating systems that eventually became us, right? And that's the RNA world hypothesis, the idea that RNA came about before DNA and before proteins, and that this is actually what eventually led to our current biology. And that sort of makes sense also if you think about the fact that proteins are made using RNA-containing ribozymes, right, and using transfer RNAs. Okay, so this idea that, and that RNA can catalyze peptide bonds. Okay, so there's definitely some evidence that maybe RNA came first, and it was really the first biomolecule that then led to these more complicated other structures that we now have. Although one could argue that RNA itself is incredibly complicated. Okay, so now I'm going to take a few minutes to get back to that question of thermodynamic stability that you asked about. So you thought I forgot, but I didn't. So really thinking about how do we actually think about thermodynamic stability. So if we think about so we're getting back to some, thermodynamic chemistry here. So thinking about our helical conformation of a duplex DNA or an RNA structure, right? You could do this with a more complicated RNA structure too. But most of the time when we're talking about this, we're usually talking about duplex DNA, right? So if you think about a duplex DNA, okay, so this is our B form, and you have all of your Watson-Crick base pairs. If you think about the thermodynamic equilibrium here, just in terms of the non-covalent interactions that are holding this duplex together, right, it can actually unwind into two single-stranded DNA, right? And so when we talk about DNA stability and the thermodynamic stability of DNA, this is the equilibrium that we're typically talking about, okay? The equilibrium between the B form of DNA, where everything is hydrogen bonded and in that helical format, or the two single-stranded DNA. Okay. Entropically, which one's more favorable? Without considering water. Yeah, this is, right? The single-stranded. Now, when you consider water, much as with proteins, the entropy of water actually does favor the helical conformation. but not as much as it is in proteins. It's actually a relatively minor contribution to the favorable binding interactions overall. Okay, so unlike in protein, as we'll get to, it actually, the role of entropy of water has a much larger contribution than it does in DNA. But there is some, right? So with water added, the hydrophobic effect does still lead you towards this. Okay, but if you're just looking at them as you know, forgetting the water, just thinking about it in a vacuum, right? This is the entropically favorable side. Okay, so how do we think about, so when we're thinking about, you know, how thermodynamically stable any particular DNA is, and I showed you, you know, a table where I talked about the base stacking interactions and we had a delta G, like how did they get those numbers? What kind of things did they use? Well, they used the melting temperature, and how did they bother to find out the melting temperature? Which I'll go into what that is in a minute. But the way that they looked at the melting temperature is by using a interesting feature of DNA, which is called hypochромicity. Hypochромicity is generally any time when the absorbance actually doesn't make sense with the overall sequence of a particular molecule, right? So when the predicted absorbance and the actual absorbance don't match, right? And one of them is lower than the other, that's called hypochромicity. When you're lower than the predicted absorbance, okay? So why am I talking to you about this? Because it turns out that the interaction between bases actually lowers the absorption of the bases of 260 nanometer light, right? So in helical DNA, the pi stacking interactions actually lower your absorption at 260. When you destroy the helicity by heating it up, right? So now you're forcing it to go or you're giving it energy to go towards the unwound form, right? It's essentially called denaturing. When you go from a helical structure to a non-helical to the two single-stranded, it's called denaturation. When you denature it, and you can do that in a lot of different ways, right? By giving the system energy. So one way that you do that, because you increase the entropy component, right? So you make the entropy of the higher by simply heating it, because then T delta S is much larger, right? You end up losing your pi stacking, and that actually causes you now to increase your absorption at 260. And in fact, even in a random coil, because you still have some base stacking, you actually have lower absorption than you would in the single mononucleotides. Okay, so if you were just looking at the nucleobases by themselves, one by one. Okay, so this change is really often used to, it is the most commonly used thing to monitor DNA denaturation, right? Going from the helix, the base paired helix, to the two single strands. Okay, so this is a denaturation curve of DNA. And it's the relative absorbance at 260. As I said, you have lower absorbance when your DNA is stacked, right? When you're in this helical format and all of your bases are stacked. As you heat this up, you'll notice that you transition to a much higher absorbance state, which is the two single-stranded DNA. Okay? If you look at this transition, you notice that it's actually in a very low, this transition happens very sharply, right? There's a very short temperature range, 30 degrees, in which you're actually going up quite quickly. That is actually evidence for cooperativity. We talked about cooperativity before when we talked about how these things zip up, right, from hydrogen bonding. And it is a hallmark of a cooperative system that you have a very fast transition like this. Okay? This, where you have 50% of your DNA melted, which you can tell because it's the halfway point in this curve, is your melting temperature. So when people talk about DNA stability and they talk about the relative melting temperatures, they're talking about the temperature at which 50% of their DNA duplexes are single-stranded, right? So where you have a 50-50 mix of duplex and single-stranded DNA. Okay? And that number is often used to compare thermodynamic stability between DNA duplexes.
So if the human body undergoes 77 degrees, and now, why are we moving?
Well, this is actually, in this particular case, we're looking for some random assigned short DNA, right? Our DNAs are not 150 base pairs, there are a couple 100,000. So we have a lot more in DNA, right? They're much longer, which means that they are much more thermodynamically stable, right? All of these numbers are typically gotten for things that are on the order of 40 to 50 bases. Okay, so most of the time when people are talking about melting temperature, that's around what they're looking at, just to get an idea of the local thermodynamic stability of different sequences. Okay, and I want to point out that denaturation is reversible. If you cool this back down, it will go to the duplex. Why is this important? It's actually really important for molecular biology. And when we talk, hopefully again, towards the end of the semester, I'm like, at the end of the semester, when we talk about things like PCR, right? The fact that you can denature into single strands and then renature it back into a helix is actually part of that cycle. No, but you have enough, and it is concentration dependent. I don't talk about that here because in general this is sort of a detail, but people actually get the numbers for delta G, for delta H and delta T using concentration.